Hello and welcome back to the Micro Lab. Today, for this video, I'm going to be going over the simple stain, which is exercise 3 in your lab manual. So we're going to be kind of going over a lot. We're going to be going over, because this will be the first stain that we will be going over. So we're going to go over what stains are. We're going to be going over what they show you under the microscope, what a simple stain can show you under the microscope. And we're going to go over, I'm going to demonstrate how to make a smear and how to do a stain, and then we are going to see what it looks like under the microscope after we are finished. So it's going to be kind of a long video. So starting out, we're going to just go over a little bit about the simple stain. So the simple stain is exactly what it sounds like. It is the simplest, easiest stain that you can do in micro. We use what are called biological dyes to do our stain. So biological dyes are also known as biological salts. So what that means is there is a positive portion and there is a negative portion of the salts. So it's an ionic bond. It's an ion. So we have a cation part of it and an anionic part of it. So we've got 3 different kinds of biological dyes that we use in here, or use in micro. We have a cationic dye, an anionic dye, and a neutral dye. Now in this class, we're not going to use the neutral dyes at all, so you don't have to worry about the neutral dyes. But we are going to talk about the cationic dyes and the anionic dyes. So remember, I just said that our biological dyes are salts, so there's a positive portion and a negative portion. For a cationic dye, the color, or the chromophore is what it's called, it has a positive charge associated with it. So these are going to be the dyes that are known as cationic dyes because they have a positive charge associated with them. The color is positively charged. In an anionic dye, it's the opposite of that. The chromophore, or the color portion, has a negative charge that is associated with it. So that's why it's called an anionic dye. Now, for a cationic dye, it has a positive charge, so it's going to react with something that is negatively charged because opposites attract. So positive is going to react with something that's negative and is going to form an ionic bond. That's the actual dyeing process. So if we're going to be using a cationic dye, we have to be dyeing something that's negatively charged. So in bacteria, their cell walls have a net negative charge associated with them. So if we are wanting to dye the outer part of the bacteria so that we can see its shape, arrangement, other characteristics, we are going to use a cationic dye. So that's what we're going to use today in the simple stain. That's what you're going to use the majority of the time, is a cationic dye. So some examples of cationic dyes are crystal violet, methylene blue, safranin, and there are several others, but those are a few that we use here in this lab. Today for the simple stain, we're going to use crystal violet, which is a purple color, and it looks really good under the microscope. So a cationic dye is dyeing the cell wall of the bacteria so that we can see the shape and arrangement and other characteristics. An anionic dye is negatively charged. So when you apply an anionic dye to your bacteria, it's not going to dye the cell wall. It's not going to dye the actual bacteria. It's going to be repelled by the bacteria. So what happens is that the glass slide that your smear is on has a positive charge associated with it. Glass has a positive charge. So when you place the anionic dye on the glass slide with the bacteria stays translucent and the glass slide itself is stained. So this is called a negative stain. Makes sense, right? You're using an anionic dye that has a negative charge, it's called a negative stain. We use this whenever we are potentially dyeing bacteria that have a capsule. So if they have a capsule, the capsule cannot be stained, so we need a way to visualize these bacteria. We use a negative stain for that. Under the microscope, it basically looks like an X-ray. The background is a dark purple color, and then purple/black, and then the bacteria themselves stay translucent. Today we're not going to do that. We're going to be just doing the simple stain today. So we're going to be using our cationic dye, crystal violet. Now, let's talk a little bit about the simple stain. It is very easy. It is one step. You make your smear. Once you've got your smear made, you dye the smear with your crystal violet. Once you're done, you rinse it, blot it dry, and then we're going to look at it under the microscope. So everything is the same color. All bacteria, no matter what kind of bacteria you're dealing with, it's going to be the same color if it can be dyed by a cationic dye. So when we do a simple stain, we do it to be able to see size, shape, and arrangement of the bacteria. Okay, so size, how big are they? Because if you're dealing with, say, a yeast is eukaryotic, so they're going to be way larger. So we're going to look at size. Shape is very, very important. All bacteria are classified based on their shape. So if you think you have some type of infection or some type of bacterial contamination, that's going to help you identify what kind of bacteria it is. And then the arrangement. That's how the bacteria are interacting with one another. Okay, so we're going to go over what these mean. So size in this class we're really not worried about because, like I said from previous videos, pretty much all bacteria that we deal with in here are going to be about a micrometer large. Maybe 2 micrometers if we're dealing with a large bacillus, but other than that, pretty much everything's going to be a micrometer large. So we're really not that worried about size in here. But what we are worried about is shape and arrangement. So bacteria come in 4 main shapes. Now, there are exceptions to these rules, but 4 main shapes. Rod is the first one, and rod is exactly what it sounds like. They look like little sticks, little rods. Now, you can have a very short, stubby rod, like E. coli, or you can have a very large rod, which is bacillus. So size, of course, is going to change. Another word for rod is bacillus. So those words a lot of times are interchangeable. So sometimes you'll hear people say bacilli, that's going to be a rod, or they might actually use the word rod. Second is cocci. Okay? Cocci is a round or a spherical shaped bacteria, and it's going to be perfectly round. They look like a little ball under the microscope. So cocci is the correct terminology. We do not use round, we do not use ball-shaped sphere, anything like that. You need to use the word cocci if you're dealing with a spherical bacteria. An example of that is staph. Okay, so we have a staph, we have cocci, we have a rod, and we have vibrio-shaped bacteria. Vibrio-shaped bacteria are also known as comma-shaped bacteria. So they look like a rod, but they have a little kink in the middle that kind of makes them bend over, and they look like commas, little commas. Okay, so those are vibrios. And lastly, we have spiral-shaped bacteria. Okay, now these come in several different forms. You can have spirillum or spirochetes, depending on the size, how long they are. Okay, you can have short little corkscrews, or you can have really long spirals. But all of those are classified as spiral-shaped bacteria. Now, in this class, you're only going to see 2 of those shapes. You are going to see a rod, and you are going to see a cocci. So those are going to be the only 2 that we are actually going to do stains on in this class. But you still need to know all 4 of the shapes. Now, let's move on to arrangements. Arrangements of bacteria, like I said, is how they are interacting with one another. And there are a few different kinds of arrangements. One of them is a cluster arrangement, and that's exactly what it sounds like, where they're going to cluster together. Another word for this is staph. So if you have a bacteria that is called a staphylococcus, then these bacteria are going to be round, and they're going to be forming clusters. Okay? And they're all pretty much going to be forming those clusters together. Next, we have a chain arrangement, where they're going to butt head to tail of one another. So another word for this is strep. Okay? So when you see a bacterial name that says streptococcus or streptobacillus, okay, these are going to be bacteria that are either going to be rod-shaped or cocci-shaped, and they are going to be forming chains. Now, some of them might not be in the chains, but you're going to see these long chains of bacteria, and it usually is pretty obvious if they're forming chain arrangements. Next, we have diplo, which is where they're going to pair up into pairs. So you'll see a bunch of little pairs under the microscope. And then you also have tetrads. And tetrads, of course, are where they're going to form fours. Okay, so you'll have them in fours under the microscope. Okay, so those are the different arrangements. Now, the arrangements that you're going to see in here, again, are only going to be two of them, is the cluster arrangement and the chain arrangement, because that's the most common, okay? And they're easy to grow. So those are going to be the ones that you're going to see under the microscope in here, okay? So now that we have went over the stain itself, now we're going to move to a workbench, and we're going to go over the steps to making a smear, aseptic technique, and actually make a stain. Okay, so now we have moved over here to a workbench, and so if you were actually in the lab, this is where you would be doing your smear, stains, working in the lab, doing everything that you need to do in the lab. So we are going to start out by going over a little bit of aseptic technique, and in micro, aseptic technique, of course, is very, very important. And what that is basically putting your target bacteria where you want it without contaminating it. Now, in micro, this is so important because a lot of times we're dealing with unknown bacteria. So we don't even know what bacteria we're dealing with. So if you contaminate it in the process of trying to figure out what it is, then you're really in trouble because you don't even know that you contaminated it most of the time. So to go over aseptic technique, we first have to go over how to use a Fireboy and a Bunsen burner. So every table in here has a Fireboy on one side, which is going to be automatic, and then we have the old school Bunsen burner. Now, I really like the Bunsen burners just because once they're on, they don't go out. So we'll discuss how to operate both of these. Now, this Fireboy is a little bit different than the other Fireboys that we have in here. So the other ones use a different system, but we're going to go over this one, and then we're going to go over how to work the Bunsen burner. So for the Fireboy, the first thing that you need to do is to make sure that your gas is on. So here's my gas. When it is perpendicular to the tube, it is off. So the first thing we want to do is turn our gas on. So you're going to put the valve in line with your tube, okay? And then we are going to light our Bunsen burner. Now, normally you would not want to wear gloves when you're dealing with the fire because you would not want to burn yourself or melt the latex to your skin. But I'm used to doing this, so I'm going to leave my glove on. You turn your Fireboy on, and then you would simply tap this, and it turns the Fireboy on. So you can see my nice blue flame, okay? And so that's what I would use to flame any of my tools. And when we say flame, we're saying that we are sterilizing it. We're killing anything that could potentially be on the end of the tool. Now, the tool that you're going to be using today for the simple stain is called an inoculating loop. So here's my inoculating loop, and you will see that it's got a little circle at the end. And that's what I'm going to use to make my smear and to get my bacteria. So if we're going to be using this loop, I need to flame it. So I'm going to put it in the tip of that inner blue cone that's inside of my Fireboy, inside of my fire. And I'm going to do it until it's red hot. You will see it turning red hot. Once it's red hot, it's sterile and I'm ready to move on, okay? Now, once this has been flamed, you don't want to touch it to anything else because now it's sterile. Do not touch it to your skin to see if it's cool. That is a huge mistake that I see all the time. Don't blow it, okay? As soon as you blow your breath all over the loop, it's not sterile anymore. So you're just going to simply hold it for 10 to 15 seconds until it's sterile, okay? And then, of course, you continue on with making your stain. Now, if you're using the Bunsen burner, which is the old school way, okay, of course, the gas is off right now because this does not control the gas at all. So it's going to just come out of the top. This controls it a little better so you can leave the gas on. Here, the gas is off unless you're actively using it. Now, to light the Bunsen burner, you're going to need your striker, okay? And in the drawers, there are strikers. The striker makes a spark so that I can light my fire. So there are little windows down here at the bottom where that controls the amount of air that's going into the fire. So to light these, the easiest thing to do is to shut the little windows, turn the gas on. You'll hear the gas come on. Hold your striker to the side, and then light. Now, I do this all the time, so I'm really good at it. If you were in here trying it for the first time, it might take you a few times, depending on how good your striker is. Now, you'll see that this flame is very wispy. It's kind of all over the place. That's not an intense enough fire to properly flame our loop. So what we're going to do is we're going to open the little windows at the bottom. Turn the gas down a little bit. We're going to open the windows at the bottom, usually about half or 75% open, and you will hear the difference in the fire. You can hear that it's more intense. There's more air going to it. And there's an inner blue cone. At the tip of that inner blue cone is where you want to be when you're flaming your loop. So we hold our loop there until it gets red hot, which I've already done this, so didn't have to do it again, but it doesn't matter. And now my loop is sterile, okay? Now, like I said, this Bunsen burner is going to stay on until you turn the gas off. So if you do use a Bunsen burner, it's really important for you to remember to turn it off, turn the gas off all the way. So we're going to turn that all the way off and move that to the side. So that's how we use our Bunsen burner. Now, for this stain, I'm going to use the Fireboy just because it's easier. I like this Fireboy here at this workstation, so we're going to use that. Okay. All right, so now we are going to move on to actually doing the stain itself. Now, the first thing that we need to do is we need to learn how to make a smear. I'm only going to go over this once because every stain that you do, you're going to make a smear for. That's what you have to do before you move on to making the stain. So I'm going to go over this in detail just this one time. And then after that, after I'm going over the other stains, I'm not going to demonstrate the stain and go over it, the smear and go over it in every single step, okay? So every table has a beaker with disinfectant and slides in it. We reuse our glass slides in here, so the first step that we need to do is to wash the slides. Now, I'm only doing one smear today, so I only need one glass slide. So I simply take my glass slide out, and I also have detergent. This white powder that's by the sink is detergent. So we're going to clean these slides really good. So to do that, I'm going to take a little pinch of my detergent, and I'm going to simply clean my slide with the detergent. So you just want to get it really good and clean. These slides haven't been used yet, so they're not that dirty, but throughout the semester, they'll get dirtier and dirtier. Okay. So we've got our slide clean with the detergent. Now we're going to get it what we call squeaky clean. So we're going to dry off some of that excess water that's in there. On there. And then we are going to use Chem wipes, which are lint-free, and alcohol. Okay, so this is going to help get any other stain that could be on there. Now you're going to get any other stain that could be on there, and then it's also going to leave the slide really good and dry, and that's what we want. And we call it squeaky clean because you can actually hear it squeak. So, now I've got my slide nice and clean, and I'm going to now place it on my staining rack. So this is a staining rack. You'll notice it's got all kind of stain already on it. So when you're dealing with your slide, you always want to deal with it on the edge, okay? You don't want to put your fingerprints or any other smudges on the actual slide itself. So we're going to handle it with our fingertips on the edges, and then we're going to put it on our staining rack, okay? So now comes the part where we're actually going to start making our smear. So there's a couple different ways to make a smear, depending on how you're given your culture. If your culture is in a broth, meaning it's in a liquid form. You simply take your loupe, flame it, dip into the broth, and then smear it on the slide and let it air dry. However, I don't like doing it this way because the broth culture does not give you enough cells with one loupe full. So the cells are very spread out and are very hard to see under the microscope. I like using solid cultures. So this is a solid culture. It's on solid media, so it's on a slant, and you have colonies that are here on the slant, okay? Those colonies, those whitish, clearish looking things. These cells are very concentrated. So we're going to get a lot at one time, so it's a lot easier to see under the microscope. So this is what we're going to be using. Now today I'm going to do what's called a mixed smear. So I'm going to do an E. coli, which is a rod shape, and I'm going to use a staph, which is a cocci. So that way we can see the rod and the cocci under the microscope at the same time. If you use a solid culture, however, we can't take the bacteria the way that it is and just put it on the slide. Because if you do that, it's going to be basically a big black blob under the microscope. The cells will be too concentrated and then you can't see anything. So we have to dilute it first, then smear it around to where it gives me a nice, thin, even layer of cells. To do that, I have to put water on the slide first. Okay? So that's what we're going to do. We're going to put water on the slide, then we'll put our bacteria. Okay, so I've adjusted the camera a little bit, so hopefully maybe you can see the slide a little bit better and see what I'm putting on here. So my slide is nice and clean. I've already sterilized my loupe. So what I'm going to do is I'm going to use my loupe to add a loupe full of water to the slide. And since I'm doing a mixed smear to bacteria, I'm going to do two little puddles. I'm going to do one here and then one close but not on top of it. So I'm going to hold my loupe under the water in the stream of tap water coming out of the sink, and I'm going to dab that little loupe full of water onto my slide, and I'm going to do the same thing with a second loupe full of water. Okay, now I'm going to flame my loupe again, because of course we don't want to put tap water into our bacteria because they're pure. Okay, now I'm going to let it cool a little bit because we don't want to burn our bacteria. If you go in and get your bacteria before it cools, you will singe them. The cell walls will break open and you won't be dying anything. So we don't want that to happen. So we're going to let this loupe cool a little bit. Okay, and I'm going to go in and get my first bacteria, which is going to be the E. coli. So I'm going to, and the cap never touches the tabletop. Remember, aseptic technique. So I'm going to take my flamed loupe. I'm going to go in. I'm going to slide right across the top to get a little bit of bacteria. And it does not take a lot. Okay, this is bacteria. Look, you see just a little bit of white on the tip of that loupe. That's all that I need. I'm going to tap this into one of my loupefuls of water. And now I'm going to flame my loupe. I'm going to flame my loupe, burn off any bacteria that's there. And you can see the little white puddle there. Okay, that's my bacteria mixed with the water. And you see how concentrated that is. We don't want that because you need one nice single layer of cells. So we're going to smear it out to dilute it. So now there's my staph. Here's my staph that I'm going to be doing. So again, I'm going to go in, do the same exact thing, go right along the top. Doesn't take a lot. A very little bit of staph goes a really long way, so we don't need a bunch. And then I'm going to smear it, put it, dab it into my second puddle of water that I have here. And notice my puddles of water are kind of close together. I'm going to mix these together. So you don't want a puddle over here and a puddle over here because you can't mix those. We want them to be pretty close to each other. So I'm going to burn off the rest of this staph on here because it's kind of a lot. And then I'm going to mix. Okay. So I'm going to let this cool a little bit because we don't want to burn our bacteria. And then I'm going to mix these two little puddles of water together to make one large smear. Okay, so now I'm going to mix very lightly because I don't want to drop my slide into my sink. That would be horrific. And you see how I'm thinning it out, okay? Making it thinner. Your book is going to tell you to make it about the size of a quarter, but if you need to go larger, you can go larger, okay? You want it to be a nice, thin, even. I know you can't see this right now, but I just want to hold on to the slide because I don't want it to fall. All right, so now I've got my nice, even smear. So you see how it's kind of clear, but you can still see that there's something there. That's what you want for your smear, okay? You don't want it to be opaque, meaning you can't see through it at all and it's milky white, because that's going to be too thick. Because before we can move on to the next step of making my smear, we have to let this air dry. All smears. When you're making a smear, they have to be air dry before you move on to the next step, which is heat fixing, okay? So if you don't let this air dry completely before you heat fix it, the heat fixing is not going to work. And heat fixing is very, very important. You'll notice in your lab manual, heat fixing does 3 things. Number 1, it makes the smear stick to the slide. You are going to be rinsing this with water. So if you don't heat fix it, when you rinse it with water, all the bacteria is going to slide off. We don't want that to happen. We want them to stick. So the heat makes the bacteria stick. Second, it creates holes in the cell wall for this, so the stain has somewhere to go. Okay? If I don't heat fix, we're not going to have open pores there, and it's going to be a little bit more difficult for the stain to get into the cell wall. And lastly, it kills the bacteria. Remember, aseptic technique. We don't want to be transferring E. coli onto our microscopes. We want all the bacteria to be dead, and that way we can help prevent contamination occurring on our microscopes or on our hands or on anything else. As long as you follow directions, the bacteria should not come anywhere close to touching you or anything like that, okay? So we're going to wait for this to air dry, and whenever it's done air drying, we're going to do the next step, which is heat fixing. Okay, so now my smear is completely dry. So as you can see, okay, it's dry all the way. It's not wet at all. So now I'm going to heat fix. So to heat fix, you're going to need your Fireboy again, or your Bunsen burner if you were using that. So I'm going to light my fire on my Fireboy. And what I'm going to do is I'm going to pass this slide over the fire 3 times. I want to warm the underside of the slide. I'm not cooking anything, so I'm not turning it upside down and cooking the smear itself. We want to do the underside of the smear. So what we're going to do is we're going to pass it over 1, 2, 3, and then we're going to touch it to the back of our hand. It should be warm to the touch. If it's warm to the touch, then I've correctly heat fixed my smear. And that's all you do. Now, you don't want to hold the slide over the fire continuously because, number 1, you're going to burn your smear. Number 2, you're going to burn yourself whenever you put it onto your hand. Okay? Also, don't be afraid of the fire. You don't want to hold it all the way up here because that's not going to be heat fixing anything. You want to run it through the fire 3 times, touch it, it should be warm, then you're done heat fixing. After you've heat fixed, you have now completely finished making your smear. So you're done with your Fireboy. You don't need that anymore. So this whole process that I've talked about of making adding the water, making the smear, air dry, heat fix, these are all the same exact steps that you're going to perform no matter which stain you're doing when you're using a cationic dye. Now, a negative stain is a little bit different, but when you're using a cationic dye and you're staining the cell wall, this is how your smears are going to be made. So now that I've got it heat fixed, I'm ready to actually move on to the staining process. Now, for this simple stain, like I said, we're using crystal violet, so it is a purple color, and these are biological dyes. So if you get them on your clothes, it's probably not going to come out, and if it gets on your skin, it's going to be there for a couple days. Now, so I always like to wear gloves whenever I do this part so I don't have purple fingers or green fingers or whatever color I'm dealing with. So in your lab manual under the procedure, you will see that it says flood the slide for 60 seconds, 30 to 60. It might be 30 to 60 or 60, I can't remember. So what you're going to do is you're going to take your crystal violet and you are going to literally flood the slide. You want to completely cover the slide with crystal violet because you want to make sure that you get the entire smear. Now, some steps, if you Google this or YouTube it or something like that, then you might see them only covering the smear. In this lab, we like to cover the whole smear no matter what. That's why we leave it on the staining rack. That way, we don't have to worry about if we got the smear or didn't get the smear. This way, if we cover the whole slide, we know for sure that we're dyeing the smear itself. So once we've done that, we're done with our crystal violet. We can put that up, and we are going to let it sit for about 60 seconds. It doesn't have to be exactly a minute, but you just want it to be around a minute. You can set a timer if you want or anything like that, but I'm just going to take about a minute. Now, after this sits for 60 seconds, we are going to rinse the dye off with water. And we're going to rinse it until the water runs clear. We want to make sure that we have all of the excess dye off, because if we don't have all the dye off, it's going to be very difficult to focus it on the microscope. So we're going to rinse our slide with water till it runs clear. Then we're going to blot it dry with our bibulous paper. Now, this dries the slide without leaving any lint or anything like that, and absorbs any of that excess dye that's on there, and that's why that it's all stained all kind of different colors. Okay, so it's been about a minute, so we're going to rinse this. Okay, and you'll see it's running off, and I like to rinse the back too, just to make sure there's not any extra on there. And you'll see how my water is running clear now. Okay, so I've rinsed it enough. I'm going to shake off the excess water, and I'm going to just pick a spot in my bibulous paper, and I'm going to place my slide in my bibulous paper, and I'm going to blot dry. Now, blot, okay? You don't want to pick this up and crunch it like that, because you're probably going to break your slide if you do that. And that happens several times in the semester. People get too rough with it and break their slide, and you just did all that work, and now you can't use it, okay? So we want to blot dry, making sure you want your slide to be completely dry whenever you put it on the microscope, because it'll stick. If you don't, it won't glide correctly if it's wet. Okay, so here's our simple stain, and this is what it looks like under the microscope. And as you can see, I have an arrow pointed right at the rod-shaped bacteria. So if you remember, we had the E. coli and staph mixed together. So we had a rod-shaped bacteria and we had a cocci-shaped bacteria. So the arrow is pointing at the rod-shaped bacteria. Now remember, a simple stain, everything's the same color. So both of them are purple, but you can clearly see the difference between the rod and the cocci. Now I have it pointed at the cluster of cocci. It's kind of hard to see the individual cells in the picture, but if you look closely, you'll see that those are more round-shaped and they're clustering together, whereas the rod is by itself. So this is a nice even smear, and this is what it looks like under the 100x objective lens with the oil.